Hey, what's happening, Rossies? Welcome back to another episode of Rossie Teaches. Today's misconception is going to be on Intro to Histology with cell staining. So there are three different types of cell staining that we are being taught in lecture. So we have the hematoxylin staining, the eosin staining, and as well as the PAS or the periodic acid shift staining. So let's get right into it. So starting off with the hematoxylin. Hematoxin staining stains acid structures blue. Okay, acid structures. So, how I want us, or a mnemonic for us to remember what hematoxin does is HBB. All right, so H standing for hematoxylin, one B standing for blue, and the other B standing for basophilic, right? And so we can see this in this diagram on the bottom left right here. This is taken directly from our lecture, where we see all the high rich acid containing structures being dyed the color blue, okay? And commonly, the cell structures that are being stained blue are DNA nuclei and as well as RNA-rich regions. Okay, so let's go over that one more time. Hematoxin stains acid structures blue. Let's remember the mnemonic HBB, H for hematoxin, B for blue, B for basophilic. Okay, DNA nuclei and RNA-rich regions are stained this blue color. Now, eosin, on the other hand, stains basic structures red and pink. A good way for us to remember this is the mnemonic ear. Okay, so E for eosin, A for acidophilic, and R for the color red. Okay, and this can be depicted here on the bottom left. Again, taken directly from our lecture, where we see commonly colored or commonly dyed or stained cell structures and the color red in the form of cytoplasm, mitochondria, or muscle. So again, eosin stains basic structures red and pink, the mnemonic ear, E for eosin, A for acidophilic, and R for the color red. And now the last staining is a special type of staining, the PAS staining, which stains glycogen and carbohydrate structures the color magenta. Now, how I would like for us to familiarize ourselves with this type of staining is the mnemonic pasta. And the reason is because if we can remember pasta, we understand that pasta is carbs, right? Which is perfect because this staining specifically comes after or goes or highlights carbohydrates or where glycogen is present. So here on the left, we have a picture of kidney cells, and two arrows pointing to the brush border and the basement membranes. Again, this is all taken from our lecture, and the reason why these are specifically highlighted is because the brush border contains glycocalyx, right, which is a type of protein that obtains glycogen, and as well as the basement membranes, which contains glycoproteins. Again, the prefix word glyco, meaning there's carbohydrates attached to these cell structures. Most commonly, PSA staining is found in kidney biopsies, glycogen storage disease, and as well as fungal infections. So we can familiarize ourselves with PSA and carbohydrates and glycogen, where we will be able to differentiate PSA from eosin staining and as well as hematoxin staining. Okay, that is all for today. Thank you, and as always, go Rossies.